Yeah, great. So I was am a cohort 3 fellow, and my project is working on the same data set that you heard from Jasmine. And I'll be looking at the non-malaria sites, looking more at the human genetic diversity and trying to find genetic variants that impact cardiometabolic traits. So next slide, please. And so our data set is a TopMed project called the Africa 6K, and it has about 6,392 samples. And this is a really unique data set to be able to study human genetic diversity, because not only does it have a large number of samples, but it has a very large amount of ethnic diversity with over 200 different ethnic groups coming from Botswana, Cameroon, Ethiopia, Guinea-Bissau, Ghana, Kenya, and Tanzania. The first part of my project is to work on characterizing this genetic diversity by using a program called Admixture, which can give us an idea of the genetic ancestry, although it's more of an approximation of the actual genetic ancestry, which is what this figure here is showing. This is from an older paper. I don't have a figure of that to show you just yet, but the idea is the different colors represent different admixture modeled genetic ancestries. And so the quantity in that pie chart would represent the amount of that admixture genetic ancestry found across different areas in sub-Saharan Africa. And we need to understand the genetic ancestry so that we can understand how we want to group different populations in our samples in our data set to be able to actually do other types of comparisons, which would be my next aim in my project, which is to run different scans of positive selection using a program called CellScan. And so this will help us identify variants that might be have more of a biological function if they're under positive selection. And the idea is that if we can look for a bunch of these different selection signals and run something like a gene ontology analysis, we might find different populations have different selection signals that might be related to cardiometabolic traits. And then the final element of my talk will be of my project will be to combine our genetic data with some proteomics, which we're using with Olink. So we'll have about 3,000 protein biomarkers, which will then be able to look at if their different levels are associated with these scans of positive selection, as well as other genetic variants in the TopMed Africa 6K. And then the final element that I forgot to mention, if we look at the top table, we've also combined the Africa 6K dataset with some of the publicly available samples in the TopMed Freeze 10 dataset, which has over 4,000 samples from the Human Genomes Project, Human Genome Diversity Panel, and the 1,000 Genomes Project to help us get a sense of how the populations in Africa compared to other populations from elsewhere around the world. Next slide, please. And so how I accomplished this project, all of my analyses were run on the 7Bridges platform. And on 7Bridges, there's a public admixture app. So I was able to use that with some slight modifications so that I can control the random seed function so that I can run a lot of different replicates of admixture for different numbers of ancestral populations. And then I also had to develop my own app to be able to run CellScan and all the different selection scans that CellScan can input. And I received a lot of help from Dave on that because I was struggling with a few of the elements since it was the first app that I ever composed for the platform. And so being able to get help from the 7Bridges stuff was very helpful. And that's all I have to present.
Thanks, Daniel. Questions for Daniel right away?
I'll ask a quick one, I guess, if that's okay.
Yeah, of course.
Hi, this is Nathan Bowen at Clark Atlanta University. So I'm interested in local admin.
Oh. Sorry, I think I muted you on accident. I went to mute myself.
It's okay. Where did I stop?
Apologies.
I heard
local.
I think local admixture or adaptation. I'm not sure.
Yeah, no,
I heard local.
So ultimately, I mentioned local admixture, but I'm curious on yours. Do you have to maintain storage for your reference populations, especially as you introduce the new ones from within Africa? And does that chew up a lot of your compute?
Yes.
Resources. Thanks.
Yes. Admixture is surprisingly very expensive to run. I've run it before, but not on a data set with 10,000 people. And so some of the larger K values are taking more than 6 days to run, so it becomes quite computationally intensive. So yeah, I guess I didn't have to do too much updating with storage, but it was something to be aware of because it does become quite computationally intense.
Yeah, thank you.
Markers to use. I see a question. So this was across the genome. So just chromosomes 1 through 22. But we did heavily filter to try and reduce the size. I think in the end, I kept any variant with a minor allele frequency of at least 5% or greater, and also heavily LD pruned the data to get the markers down to about 1 million. And even then, it still is quite computationally intense.